Meet the bluegill sunfish. It will eat nearly anything it can and breeds prolifically. This devious little guy ventured from Gutenberg, Iowa, to the shores of Japan, terrorizing local fish and causing the native freshwater mussels to nearly go extinct. To try and stop it from wrecking the local ecosystem, Japanese scientists had to first find where these fish were. The ocean is a huge place, and it's hard to know where to begin. These fish didn't have footprints, and any droppings could be easily dispersed through waterways. Camera traps, set up by researchers and left alone to take pictures of passing animals, were expensive, and only covered a section of the water. And so was sending people out to try and visually identify them. Here then comes our hero, environmental DNA, or eDNA for short. This is DNA found in sediments, water, and virtually anywhere else in our environment. Researching with it is quick, low cost, and efficient, and was a research method used to detect the bluegill sunfish in Japanese waters. Seems too good to be true? Let's hear about how this superpower works. Scientists simply scoop up the sample in a syringe. They then isolate the eDNA from the rest of the sample with a filter. After this, the scientists copy the eDNA in the sample through a process called PCR. In this reaction, eDNA is put into a machine called a thermocycler, which heats up the DNA strands so they split apart. Once they split, primers, which are short segments of RNA, are added to the sample. These primers then bind to regions of interest on the DNA strand, which scientists call barcodes. Barcodes are sections of DNA that have unique sequences for nearly all species. Once the primer binds, an enzyme can then replicate the barcode over and over. Just 20 to 30 cycles of this can make millions upon millions of copies of DNA. Once enough of the DNA of interest is duplicated, it can finally be detected by a sequencer machine. This machine takes the genetic code of the DNA sample and compares it to a database of other organisms. Running through these genetic sequences, the sequencer will find a match, and boom! With just a single water sample, you can know whether the bluegill sunfish was really there. But this isn't just a story about the bluegill sunfish. Rather, it's about eDNA and the many different ways in which we can use it. eDNA has been used not only by conservation managers eager to protect their habitats from invasive species like the bluegill sunfish, but also to detect the presence of endangered species like the sawfish and the Mogollon skate. It's also been employed by fishermen and those in aquaculture who farm fish and other seafood. With just one sample, eDNA can tell fishermen if any diseases are in the water, and even how many fish are in the given area. This can save fishermen from selling unsafe food and help them better understand how much to fish in a given year, such that the fish population isn't harmed. While this is a new and budding technology, its applications are far and wide-reaching. eDNA can look at virtually any genetic material, whether it be the hair that falls off of our head or the scales that a passing fish can shed. The samples are also really easy to collect, and people have already begun to capitalize on that. Because of the simple sampling process, eDNA collection has been highly used in citizen science, a type of science that allows non-specialists to come and collect data. Scientists can then group source this data to get even more powerful insights that span many different geographical locales. Even high schools are practicing collecting eDNA samples. The Urban Barcode Project is a science program that spans 5 boroughs of New York City. Students sample biodiversity in local parks and gardens, check for invasive plants, and monitor animals that may be carrying diseases. Through this organization, students in K-12 education have looked at the biodiversity of ants in their homes to the types of oysters that live in the East Hudson River. In fact, through the Urban Barcode Project, the area now has more than 500 newly identified species. Want to get involved? It's not too hard. Scientists and organizations like Cal eDNA are looking for help. By joining their team for free and getting trained online, you can help collect data which can then be sent to their labs to process. Who knows? Maybe you'll discover the next new species.